Hello, everyone. We're about to start the last contributed talk session, and we've moved the order of talks a little bit just so that you guys don't get confused. So the planning with others talk will be our first talk, and then everything else will be in the same order as in the program. So the first talk will be presented by Nastaran Arfai, and this is work with Weijima, planning with others in mind.
Hi everyone, as promised, my name is Nastaran Arfai and I'm a PhD student in Weijima's lab, and I'll talk about planning with others in mind today. So there are many factors that make us the humans that we are, but one of the features that allows us to achieve objectives so complex that we ourselves don't believe we have achieved them is our ability to plan collaboratively. And we do that on different kinds of tasks, and sometimes it can be easy and effortless, and sometimes it can be difficult and prone to violence. And these are just a few examples of brawls that have broken out in houses of representatives along the world, proving that planning with others can be a difficult task. In cognitive science, collaborative decision-making has been studied in multiple perceptual paradigms and with tasks with small state spaces. Also, some really cool work has been done on the process of inference of the beliefs and goals and desires of others, and also on how people model and predict actions of others based on such inferences. What we aim to explore is collaborative complex planning, which roughly sits at the intersection of all these three areas. So we want to see first whether people take the future actions of their partners into consideration when planning collaboratively. And then if they do, how do they model others, and how do they incorporate these action predictions into their planning process? And so to do this, we have designed a new paradigm called the collaborative construction task, where two players start from two different locations on a map, and they connect to cities or dots on this map and trying to maximize the number of cities that they have jointly caught. So let's go through an example trial here. You are the green player and your collaborator is the blue player. Your collaborator makes the first move using the shared budget equal to the amount of distance that they traveled, and now it's your turn to make a decision. You take the next move and your score is increased by 1. Your joint score is increased by 1 again. Your collaborator makes another move, turning the joint score to 3, and now it's your turn to make a decision. But it seems like all the potential actions that you can take are not better than the ones that your collaborator can take. So you skip and you decide to give that turn to the other player. And so on and so forth, the game goes on until no more actions can be taken, the trial is over, and your score is the number of cities that you collected together. So far I've described the multiplayer version of the task. We also made a single player version where one person would control both the green and the blue player. And we had 30 dyads come into the lab and play this game in 2 sessions. And people could not talk to each other during the game, and they were under no time constraints. Let's look at some data now. So we define performance as the ratio of the actual number of cities collected compared to the maximum that could be collected. And people kind of perform well. Dyads do okay on the task. And critically, they perform better than the performance of the solo optimal policy, which is the best performing policy that does not at all account for the future moves of the collaborator. which is a piece of evidence that people do consider future actions of others in their planning process. Another piece of evidence is skipping. So here we have skip rates over the optimal contribution gap, which is the number of cities that can be collected by the self minus the other if both players play optimally from this point on. And if people were not considering the other player at all, they would never skip. Or if they were skipping randomly, this would be just a flat line somewhere. But this is what we see from the data. And it shows that people skip less when their state allows more contribution to the shared score, and they skip more when they can contribute less to the shared score, showing that they're aware of the state of both themselves and the other person. Now for modeling. One of the easiest models that you can make is the greedy model, where the agent just goes to the closest city with some noise or skips randomly. And this can do well sometimes, but not always, and it can make some bad mistakes. Another very similar model is the jointly greedy one, where it's very similar to the greedy model with the exception that it also looks at the greedy action that is available to the other agent and skips if the other person has a closer city available. And that can also sometimes not perform very well. The solo optimal model, on the other hand, has access to the maximum number of cities that are reachable, that are achievable from any of the cities within its reach, and goes with some noise to one of those cities. And as you see, sometimes it does well, especially when the optimal contribution gap is positive, but not so very well when the contribution gap is negative. The tree search algorithm uses a heuristic value function to estimate the value of each state. Then it gradually improves the accuracy of the value estimates by expanding nodes of a decision tree and then backpropagating the values down to the root of the tree. In its collaborative context, an agent can evaluate possible gameplays by interleaving its own actions and the predictions of the actions of others. So we built a model using this approach with a value function that is the linear combination of the number of cities that were caught, the number of cities within reach, the remaining budget, and the value noise. And then it does a best first search with a given stopping probability and a lapse rate and a pruning of the unlikely branches. But the potential problem with this interleaved approach is that it requires people to take their attention back and forth continuously from themselves to the others, and that might not be something that people actually do. So we made an alternative model where the approach is to create parallel tree search models, starting from the current position and doing a separate search for the self and the other, deriving the value of actions available to the self, but also the value of skipping as the highest value action available to the collaborator. So how do these models do? We fit each model to the behavior of each individual subject separately, and plotted the total AIC and BIC of each model across all participants minus that of the best performing model, which is the parallel collaborative tree search model here. We have also used the best fitted parameters from each model to each subject to simulate actions in these various states, and we'll overlay model results on top of the data. So here, accuracy, we are showing accuracy as a function of optimal contribution gap and optimal depth. And this is the results from the greedy model, jointly greedy, solo optimal, interleaved tree search, parallel tree search, and here parallel tree search alone. We do the same for skip rates. And we see the greedy model, jointly greedy, solo optimal, interleaved tree search, and parallel tree search, and here by itself. So in conclusion, we showed that collaborative planning is an interesting area of cognition that is also a bit understudied, that people can account for their collaborators' future sequences of action during planning, and that a parallel collaborative tree search model performs the best so far in predicting people's behaviors. we will, so far we've been using the same value function for the self and the other, and our next step is to use different value functions for these and improve the tree search models. We will also look into the dynamics of eye movement and how they correspond to these models, and also we will look at communication during collaborative planning. I'd like to thank my advisor, Wei Jima, and the wonderful MALab, and thank you for your attention, and please reach out if you would like to continue the conversation.
Questions? Time for one or two quick questions? Come on, junior scientists, you know you have questions.
Hi, nice talk. I didn't really understand the difference between the parallel and non-parallel tree search model. I mean, is it literally you're saying that people are doing both at the same time? Like, how would you predict the attention to vary, for example? You said there would be this cost of switching back and forth. How does that play out here?
Yes. So the difference is that in the first one, you have just one tree, and that tree is looking at the turn of the self and other and interleaving that. So each branch is self, other, self, other, self, other. And that is the real structure of the task, right? This is a turn-taking task. The parallel model is not exactly what the format of the task is, but it's an approximation which builds two separate trees and differentiates the actions of others from the action of the self more, and then only compares those things. So yeah, they're pretty different actually in their function. They can start to kind of like do similar behaviors, but the underlying structure is very different.
Thank you.
And quick question, and while we transition to computers.
Great talk. I just wanted to ask if you thought, in addition to tree search, about maybe some alternatives like the successor representation or something like that?
Yes, that is something that we will look, especially perhaps on like Monte Carlo searches and those. Yeah, we will do that as comparison. But these are the models that we thought were most likely to correspond to what people actually do.
This is supposed to just, sorry, just.
Was there no cord to? Yeah.
Oh, right.
Yeah. Okay, so next up we have a talk that's joint work by Xiaolang Lu, Robert Mock, and Brad Love, and Robert Mock will be presenting, and the talk is called The Inevitability and Superfluousness of Cell Types in Spatial Cognition.
Wait, I can just mirror this, please. Oh, come on. Different port, you said? Oh, you mean different here? I don't know which one's better. Okay, great. Cool. Great to be here today. I think it's been such a great conference. I'm speaking on behalf of Xiaolang Lu, who couldn't be here today, but really spearheaded this work. I'm going to talk, this talk I'm giving hopefully will be thought-provoking. It's quite a simple message, but I think maybe we can think a bit more about how we do neuroscience. So first, how do we try to understand the brain? Historically, people had limited methods. They had one electrode, they poked their electrode into the brain and tried to find something interesting, right? Makes sense to do it that way. And we tried to look for cell types, right? This is normal in biology. People found spatial cells, concept cells, mirror neurons, you name it. And so I have a few cells here, like most people will know. There's place cells up in the corner there that look like they encode space or a location. And these are cells in the hippocampus in the rodent where the rodent might move around in this square environment and it only fires at that particular location. And so probably it's encoding a location, right, or a space. Below that are grid cells. They tile the space. It might be some kind of useful metric for space. And on the left there is a head direction cell telling you where you're going or where you're facing. And so the idea here is that there's some kind of cognitive or spatial map of the environment, and you can use these cells, combine them to navigate or understand space. So it would be nice if the brain was so simple, right? So maybe the brain is a little more complicated. So one thing we can think about is the cells that are identified are, there are some criteria that were developed based on what we found. So again, historically, you find a cell, you think, oh, this looks interesting. Oh, but at the same time, we need to quantify this a bit. It looks like it's encoding space. Maybe we need to find, how higher firing rate is important. It's not firing everywhere. It's not firing only in a tiny little place, that kind of stuff. But then this is binary, right? And neurophysiologists know this. It's not like, oh, there are place cells and there are non-place cells. There are place cells and there are more messy place cells and there are multi-field cells and so things that might not even be consistent over sessions or over even within the session. And the worry is that if we're only guided by intuition to find these cell types, we might find things for the wrong reasons. And so if we force our intuitions onto this complex system, it might be easy to fool ourselves if it looks exciting. And of course, as I said, it makes sense to start this way. We had one electrode, for one session, we can't just record the noisy cells and figure what's going on, right? So, but what if finding cell types is the wrong approach? And the worst case that we see is that it might slow progress and threaten the future of the field. So in one example from our previous work is that spatial cells are not just spatial. In this case, we found that a clustering model that can cluster concepts, in this case, hopefully you can see it's birds at the top and mammals at the bottom. If applied to navigation on the right there, creates these grid-like patterns. So it's the same algorithm for clustering concepts and space, and you get different results. And so this means that grid cells in this context is one case in a spatial context that creates this pattern of activity, whereas in other contexts it would create a different one. So there's not only us, of course, there's a lot of work now talking about non-spatial aspects of hippocampal formation representations. So our questions in this project is, are spatial cell types really cell types with specific functions? And how can we test this? So our approach, we used a VR plus a deep neural network approach. So on the top right there, you see the top-down spatial view of the arena, square environment where the agent or we put the neural network in there, kind of, and then it's just looking at the environment from a first-person point of view. On the left there, hopefully you can see that in the middle is the whole environment, and each of the images surrounding it shows that first-person views. So one thing to know is that this is a null model, right? So there's no spatial knowledge, there's no learning and no plasticity. We're only using these neural networks, which are either trained on object recognition or not trained at all. And if these networks show spatial cells and spatial information, what does that mean? And what we think it means is that finding something that looks like it's spatial or spatial cell, it doesn't mean that it is for that function. And we want more rigorous tests for function. For example, are the units actually involved in the task? So looking into these networks whilst they're kind of moving around, we do, in fact, find a lot of spatial cells. So these are only a few examples from VGG16, but they kind of exist across all layers and all these networks as well. And these are using the criteria that has been used in neuroscience literature to find place cells, head direction cells, and border cells. do these networks actually have any spatial information in them? So to test this, we use the linear decoding approach to test for spatial information in these networks. So on the top left, there is the top-down view, and then here is the kind of setup, right? You see the network sees in first person, creates a set of activations in the deep net, and then we want to say, for each layer, is there spatial information there? To do this, we sampled for location, for example, where they are. 30% of the environment, just standing around, looking around, and then we tested on 70% of the rest of the data, and said, can this decoder tell us where we are? And the same thing for head direction and the distance to border. And so what you see here is for all networks tested, trained and untrained, we have very good location decoding, far below chance, and the two measures of chance shown up there with the 2 lines. This is true for direction decoding and nearest border decoding as well. So the next question is, what's driving this? Is it actually the spatial cells, identify spatial cells that are doing the hard work? Turns out it's not. So we've excluded, based on these criteria, the top place cell activity, top border cell activity, top head direction cell activity for each decoding procedure. And it doesn't look like they're important. And we did some more analysis that's in the paper now, and we can show that there are important populations. So this is brand new work where we show that we can also find some remapping effects. So you can see that on the left there's the original units, firing fields, and there's some remapping. So at the top there's units that don't change too much, and the bottom some that actually remap in a random fashion, like in the hippocampal formation. Here's another example where it's from original to a large change, so the whole environment changes. You also have some units that show similar things and others that remap. So the idea here is our intuitive interpretation of a cell's function, like representing space, might not map onto a cell's actual function. Finding spatial cell types in these non-spatial networks, both trained and untrained, doesn't mean that they're fundamental to spatial adaptation. So are these functional cell types real, or at least sensible to study? They might be, but you don't know until you critically assess it. More importantly, for me, I think there's a lot of cells. It's not just, if someone has identified a cell with a criteria and said, this is important to us and published it, then the whole field, or a large proportion of the field, might just look at those cells. And I think that's could be worrying, because there's many cells that may have unusual properties, multi-mixed selectivity, and are ignored. So basically, we call for a high bar for these claims about function, and our example is a model that can serve as a baseline for it to be surpassed before making strong claims. And it's kind of like a null hypothesis test or a permutation test in statistics. So yeah, that's basically the message. And I'd like to thank you all for listening. As I said, Xiaolang did the hard work, and on a totally different topic. I'm going to be moving to Osaka, Japan next year, and if anyone's interested in postdocing or PhDing on category learning, memory, task recommendations, we have lots of scanning and GPU, CPU compute. Thank you. And come talk to me if you're interested.
Very cool and provocative. There must be questions. I have questions, but I don't want to leverage my... Are you coming down to ask a question
Yes.
or bathroom?
Yeah, I think you.
All right, so we already spoke at the poster, and I agree with the general idea that there is this set of neurons that are, very large number of neurons that are showing mixed selectivity, that are showing responses in between these strict categories in terms of a continuum of different responses. So I agree that there should be more presentation of this distribution of responses as is starting to come out in the papers. But just to fight, for my side a little bit, because I am a believer in the functional importance of these cell types, I guess one argument I would make is that I personally would not want to have the kind of the baseline null hypothesis be, a feed-forward multi-layer neural network, even if, it's a completely untrained network, because that, my perspective on the function of the brain, which, contrasts with many of those types of models, is that the feedback connections are very important. They're literally, 80% of the neocortical connectivity is feedback. The recurrent dynamics are tremendously important. So I just want to emphasize that, I agree that we should present the full range of data, but I don't want that model to be the null hypothesis.
So I think, thank you. It's important that the null hypothesis not be the right model, right? So the point here is that we can actually create these kind of cells even though it's not recurrent, it's not like the brain. I would say, so it is a feed-forward model, so it can't do a lot of things, right? It can't do sequences, and so you can kind of like say, oh, this is when we're doing our sequence learning or something like that, this is not important. But then the problem is when you're come up with criteria and say this is an important thing for this particular function, and I think, as we've discussed, like neurophysiologists know the subtleties and stuff, but I don't think I've met anyone who is not directly in the field that knows of this problem. And I think it is true for, like big papers, I found a new cell, and we should do both, right? Thanks.
Okay,
thanks for starting.
If it's a quick question, then while we're transitioning.
No, just real quick. Do you guys have data on like how robust these results are to changes in parameters, and like specifically the first thing that comes to mind is like how wide the neural network is, because then and also the threshold at which you pick the place cells, because I imagine the lower is the threshold for how specific these place cells are, then like more cells would be picked. So yeah, do you have plots showing these results standing over these varieties of parameters?
So what was the last part of that? The results that
have
were
plots showing
quite wide cells, you mean? Like not very specific?
Yeah, but like the wide or the threshold into which you choose like that something is a place cell.
Right. I mean, the why something is a place cell is defined by like various metrics, like so we followed a few papers, like this one particular paper, but then it's like firing rate above the average of the field. It's not firing over 50% of the arena. That kind of criteria. So those are the specifications. Is that?
Yeah, we can talk more later.
Yep.
But,
and then the second part is.
I think we're
okay, sorry.
Yes,
I can totally leave it.
Take it offline. Thank you so much. Okay, next up, yes, thank you. Thank you, Robert. We have Jie-Yu Zhang from Caltech, from Marcus Meister's group, presenting on mice in the Manhattan maze.
Can you hear me okay? All right, hi, I'm Jie-Yu from Meister Lab from Caltech. Today I'm presenting some work about a mouse exploring a maze, and we study their complex cognition. So mouse live in underground burrows, and sometimes the environment will change. This is similar to you living in a city setting, and there are construction sites on your daily commute, and you somehow have to detour. So the essence of a city grid, such as the Manhattan Island, is that there are vertical and horizontal streets that are dictated by the junctions, so you can make the decisions. And we utilize this feature to design the maze called the Manhattan maze, separating the vertical and horizontal streets into 2 layers and have a mask in the middle. With the holes, the mouse learns to cross the layer and go up and down in this 3D maze. So this is the top view of the maze. And we place 2 water ports at the outskirts of the maze and the home cage of the maze. The mouse learns to alternate between the 2 water ports and collect rewards. And we can then easily switch this mask to a 9-hole mask that is a lot more complicated, with just an easy control by swapping the mask in the middle, and that would make the configuration change. So how did the mouse learn the 9-hole mask? You can see that this mouse is doing very fast. Its optimal trajectory is actually just 30 seconds, and you can see how similar it is to the optimal trajectory in theory. And we can look at an aggregated data of 15 mice. And here I'm showing a learning curve based on the duration of traverses, or how long it takes for the mouse to travel from one port to the other. And you can notice that the improvement is 5 to 6-fold from 200 seconds to just 30 seconds. And I want you to notice that there's the first phase, which is the first 2 traverses, that there's a 2-fold improvement in the first homebound traverse. This is a travel that's coming from the outskirts to the home, and the mouse has never done this direction before. So once the mice have learned one mask, we can look at how they can learn multiple masks. So on day 2, we presented 2 new masks in the red and green. So on day 2, in the early sessions, the mice started repeating the same mask, basically started off the same level of performance as the end of day 1, and they saturate very quickly. This shows an overnight memory effect. And for the new masks, you can see that the learning curve is a lot steeper, and after 6 traverses, it's not differentiable from the old mask anymore. This shows the effect of generalization or continual learning, which is basically the mouse learns to accelerate its learning in a new environment. So after the mouse has been exposed to many different mazes, in the later sessions, the performance in the old mask and the new mask are almost exactly the same. There's no difference between the curves. This suggests that there's a generalization effect just over learning new environments. So after testing these mice in this mask, we tried a more complex one, basically 2 highly densely connected regions with a bottleneck in the middle. The mouse need to remember the bottleneck and navigate to the ports. You can see that this mouse improved a lot. In the first traverse, it took almost 500 seconds, but just the first homebound travel, it's only 20 seconds. And later, after just 20 rewards, it settled down on the 30 to 40 second scale. And the learning curve is pretty much the same as the previous mask that I showed you. So we know that for humans, a lot of the complex cognition depends on our cortex, and we believe that cortex is essential for all of these. So what about the role of cortex in mouse navigation? To answer these questions, we basically developed a genotype of mice that basically they were born without neocortex or hippocampus. So how did the acortical mice do in the mazes? The acortical mouse took almost 3 times longer to solve the first mask. And you can see the first reward took quite long. And this is because the acortical mouse is basically scanning between different corridors, the same corridors, and they're dropping out. Oh, I see. Okay. It's basically repetitively scanning the same corridor. And you can see that in this trajectory that the mouse is basically scanning at around the same distance. But after the first reward, the mouse has a very steep learning curve too. And you can see that it settled down at the same level as a wild-type mouse, taking only 30 seconds to solve it. So here I'm showing another video clip of the mouse solving the maze, even though it is only acortical. And an acortical mouse can learn multiple masks as well. So I'm showing here the new mask 1 and new mask 2 and its best performance. It's also at 20 to 30 second level. And for the complex mask, it could also solve it in relatively short time. And if you remember, this is almost exactly the same as the shortest path possible in theory. And for multiple acortical mice, the learning curve also improved from mask 1 to mask 2 and 3. For me, the most surprising result is that acortical mice also have long-term memory. So here I am making one mouse repeating its mask over multiple days. On first day, its performance is okay, but on second day, it continued its performance and improved. After a week's break, the performance gets even better. And so the mouse went on a vacation for a month, and it still continued at its peak performance. So I gave it another month of break, and the performance is still at the same level. So in summary, we find that the mice can do rapid learning, overnight memory, and meta-learning in the Manhattan maze. All of these results have very high effect size and with large difference. And we also find that all of these cognitive functions do not strictly require cortex. And with that, I would like to thank my team and all their contribution to this project, and I am ready to answer your questions.
So when it's come, it's great work. Thank you for your presentation. And when it's come to the grid-like maze, some previous literature says either rodents or primates can do that based on memorizing the order of the direction. Either it's like left, right, right, left, or like a sequence of orders to move. I don't know if you control the, when you switch to the type of maze in the next day, have you controlled the order of the direction? For yeah.
Okay. Yes, I want the slide, sorry.
No, it's
yeah. So thank you for that question. I clearly paid you $100 to ask it, so I can show you the slide. So basically, the new mask that you saw has two different turn sequence from the beginning. There are 9 holes. So for the red one, it has exactly the same turn sequence as the old mask. And the new mask C, it's green, and it has a different turn sequence. And we find that with different measures, there is not too much difference between the performance in the 2 different type of turn sequence. So from our observation, it seems like the mice haven't done any sequence learning.
Thank you so much.
Hi, great talk. Thank you. Very stunning.
Can you speak up?
Very stunning results. What the acortical mouse? What else? What other resources does it have to work with? How does its brain look like?
Okay. Clearly, also paid you $100 for that question. Here is a basically an MRI scan we did on the acortical mice. So in summary, these mice do not have the dorsal forebrain. They don't have the dorsal, the cortex, prefrontal cortex. They don't have motor cortex. They don't have primary visual cortex. So the remaining structures are the subcortical structures, such as superior colliculus, for example. That's integration of motor and vision, and cerebellum, which also supports learning. And there are also many different candidates. So this is a question we're still exploring.
It almost looks like it expands to the same size. Is it expanding to the same size?
No, the subcortical regions are staying in the same size. You can see the cavity over there. Basically, it's the same amount of volume. We didn't find any volume difference between the subcortical structure in the acortical mouse versus the wild-type mouse.
Sorry, we're going to have to move on. Thank you so much. That was really great. Sure. I mean, but we're going to be switching.
Right,
we can't use the computer.
Right.
Okay. Okay, so next up, we're going to have Sarah Jo Bendito presenting work with Kevin Miller, Nathaniel Daw, and Carlos Brody called Planning in the Hippocampus. More hippocampus coming up. Linking actions now comes to guide behavior.
Hi, everyone. Thanks for sticking around the last talk session of the conference. It's been a lot of fun. And I'm here to talk a bit about my project, Understanding the Role of Hippocampus and Underlying Model-Based Planning. And so just to start out, what do I mean by planning? And so planning, I am using it to refer to as using an internal action outcome model to guide behavior. So imagine you're chilling at home and you're like, wow, I would just love some pie right now. It just sounds amazing. And so in Princeton, you've got a couple options. You could go to Wegmans or you could go to Whole Foods. You don't really know who's got some good pie, but you're gonna pull out your mental GPS, because this is pre-cell phone era, and you're gonna track your course mentally, of course, to Wegmans. We're gonna just try there first, because we like going left. But as we're kind of driving down, we find that the only entrance into Wegmans is closed, forcing us to veer down Rare Street instead, and now we're stuck going to Whole Foods. Fortunately for us, Whole Foods has some amazing pie, and it's so good that the next day when we're gonna go back for round 2, we're gonna instead plan our route to go directly to Whole Foods. And so we believe that this mental GPS kind of represents this internal action outcome model that represents not only the states of the world, but also the connections between them. And we believe that this kind of exists in the brain, and we can use this type of model to propagate values from outcomes that we've experienced back to relevant choices. And importantly, having this type of model allows us to learn about actions we might not have taken. So for example, we can learn about Right Street after getting some delicious pie from Whole Foods, even though in reality we went down Left Street to get there. And so we have a pretty good reason to believe that this exists in hippocampus, and we don't have time to go through all of those reasons, so I'm just gonna flash them on the screen. But the most important one for me is the fact that it's been causally implicated in model-based planning, and specifically in the tasks that I'm going to be using in the study that I'm talking about today. But the question still remains, what are the neural representations, and how do they support this process in the hippocampus? And so I'm going to try to briefly cover this entire project in another 7 minutes by going into 3 parts. First, I'm going to go over the task itself, so hopefully you can believe me when I say that the rats are planning. And next, I'm going to show you kind of what's going on in the hippocampus, and then finally, using those representations that we see, how is that linked to planning on a trial-by-trial basis? All right, so starting with the task, we have what's known as the rat 2-step task, and this was developed by Kevin Miller. And essentially, we've got a rat that's in this behavioral chamber with 6 ports that correspond, each row corresponds to the first and the second step. So the rat has been trained to poke into these ports that light up. So at the beginning of a trial, he's gonna poke into that top center port, and then that's gonna cause lights to turn on into the adjacent 2 ports. And this is the only free choice in the trial. He can choose either left or right. And after he does so, there's going to be a sound that plays, and the sound that plays is gonna correspond to one of the 2 bottom side ports, and basically which one's going to get lit up, which grocery store is going to be open at the end of the task. And that light that will shine is dependent on the choice that the animal makes at the first step. So if the animal were to make a left choice, there's going to be a high probability that that bottom left port's gonna light up, and a low probability that the right port's gonna light up. And we're gonna see the opposite true if the animal chooses right instead. And we call those high transitions the common transition, and the low probability the rare transition. All right, so that bottom center light is to initiate the second step, so he's gonna go and do that, and then of course there's only gonna be 1 light that turns on in the outcome port that corresponds to the sound he heard earlier. And after going there, there's gonna be some chance of receiving reward. And specifically, there's going to be 1 port that has a really high chance, and the other port's gonna have a lower chance. And that, the better port is gonna kind of flip unpredictably throughout a session, and it's the rat's job to figure out, which port is better, which one's got that better pie. And they can do this. And unfortunately, we don't have time to convince you, but you can go to Kevin's 2017 paper to see a more convincing argument. But lastly, I just want to point out some notation that I'm going to be using for the rest of the talk, where I kind of refer to each of these ports with a letter and a number, the letter being the side, left, center, right, number being the row, 1 or 2. All right, so let's see what's going on in the hippocampus. To answer that, I bilaterally implanted NeuroPixels 2.0 probes targeted at dorsal CA1 of hippocampus. And I did this in 4 rats, and this allows us to really densely sample many, many simultaneous neurons in CA1, allowing us to really capture a relatively comprehensive picture of the model that's being represented. And so if you recall, we've got kind of 4 trial types that are defined by the choice side and the outcome side. And what we can do is we can unravel these port trajectories into just like this linear space. And this is what we're going to use instead of this 2D port space to kind of look at how the hippocampal units are encoding each of these trial types. So zooming in on the left trial, what we find is that we've got subpopulations of units that really like specific port events. So this top green, this lime green portion really likes that top center port. We've got another purple population that likes that top left. There's a population that likes the top right that's silent during this trial. Another population that likes the bottom center. Another bottom left, and of course bottom right, which again is silent in this trial. And so when we look across all the other trials, we can see that all the activity is timely, or we have a very comprehensive coverage of all of these trial types. What this point of view doesn't exemplify is the fact that we have even more specialized coding happening during the second step of this trial. And specifically, we've got units that really like to fire, at specific locations, but only when a certain condition is met. And for this circumstance, that condition is a choice that the animal made previously. And so this first example unit is one that fires at that bottom center port, but only after making a left choice. Similarly, we can see example units that fire at the bottom outcome ports, but only after a preferred choice is made. And this basically gives us this joint decoding of choice and state throughout the trial, and gives us kind of a distinct activity map for each of the trial types. And essentially, what it also gives us is a choice decoding basically anywhere throughout the task space. And this is the kind of decoding that we're going to look at in between trials to see how it links to planning. All right, so lastly, what we want to see is that the hippocampal activity should reflect the planned action. So what the planned action is can be kind of hypothesized from the choices that the animal makes in behavior. So I didn't show this earlier, but now I will. This is just kind of how the animals respond to transitions and rewards on subsequent trials. So for a common transition reward, animals are going to be likely to make the same choice. After a mission, they're going to be more likely to switch choices, and this pattern of behavior is true whether or not they're using a model. But we see this pattern flip for these rare transition trials. And so after a mission, these animals are more likely to stay instead of switch, and after a reward, the animals are more likely to switch. And this is kind of how we know that they're using the model on a behavioral level. And so what we want to see on a neural level is a similar pattern. And so we're going to kind of look for this pattern of choice encoding by predicting like what choice is being encoded by the neural population given some window of activity. And this is just using a Bayesian decoding approach that's used a lot in the hippocampal literature. All right, and since we only have 2 choices, we can also condense this to a single metric, and we're going to have it with respect to the same action that the animal had just made minus the opposite action. And so what we want to see is that for common rewards and rare emissions, we should see a more positive value representing the same choice, and we want to see a more negative value biased towards the opposite choice for these common emissions and rare rewards. All right, and so we're going to look for this during the intertrial interval because the animal is most likely to plan after receiving some outcome and before making another decision. And so we're going to actually, limit this a little bit further so we know exactly where the animal is and that he's engaged in the task. And then what we see during the average activity limited to these 2 windows is this exact same pattern that we see in the behavior. So we see this kind of more positive effect or more encoding of the same action after these common rewards and rare emissions. And then relative to the same transition type, we see it biased towards the opposite choice following common emissions and rewards. And importantly, this is true regardless of what the animal does on the next trial, which is really reflective of some model-based credit assignment or planning process that's happening, and not just a selection process. All right, so I think that's the last of it. I hope I was able to cover it adequately. And with that, I'd like to thank my advisors, Carlos and Nathaniel, and of course, Kevin Miller, who's been a very invaluable collaborator, as well as my labs. And if you weren't able to make it to my poster, Kevin has posted it online, so feel free to go scrutinize it. But there are some typos that I might fix before you get there, but also might not. Okay. That's fine.
Super quick, very quick. Go.
Yeah,
Let's switch.
I'm not a neuroscientist. I can play one on TV, but I was wondering, did you try, can you, with the NeuroPixel, can you knock out these cells and see that it's causally involved in the behavior?
Not with the probes themselves. I don't think they allow enough current to actually ablate anything. But the study that Kevin did previously with rats in the same task was a causal manipulation where he infused mucimol into dorsal hippocampus. So we do have that kind of causal knowledge already, just not in these 4 rats.
Thanks very much, Sergio. And last but not least, we have Erica Bush from Nick Turks-Brown group at Yale, talking about learning along the manifold of human brain activity via real-time neurofeedback.
Cool. Thank you so much for being here and for sticking around for the very last talk. I'm Erica Bush. I'm a PhD candidate in Nick Turk-Brown's lab at Yale University. So as humans, we can acquire diverse skills with astonishing speed. So for example, a native English speaker learning Spanish as a second language might then easily acquire French or Italian, given the common structures that underlie Romance languages, but they might later flounder with learning Mandarin. So why are there some skills that are easier to learn than others? There are mechanisms of neural population coding that actually relate to this type of learning, so that the functional correlations of a neural population can constrain how easily these populations can reorganize and learn new information. And these properties can be changed by what we know, such as already knowing Romance languages or extensive training. And the idea that neural co-fluctuations, or neural activation manifolds, as I'll refer to them, constrain learning has been tested in the brain-based prosthetic literature. So in this study, a monkey learns to control a brain-computer interface in order to make the cursor move over to the green dot. And in general, within a BCI framework, experimenters would take this BCI mapping and directly map the recorded neural activity onto a given feedback signal as presented here. However, they've also found that by learning and basing feedback off of a low-dimensional manifold representation of the measured activity, BCI learning can be sped up and it can be made more stable. And this can actually also highlight components of that activity which are easier to learn how to modulate. So based on this work, we'd hypothesize that the manifold of brain states can be harnessed in order to accelerate human learning. And neurofeedback is a tool that can be used as a type of non-invasive brain-computer interface to test hypotheses about learning in the brain. And despite neurofeedback's therapeutic promise, many studies have shown variable or limited learning effects, but prior work hasn't considered how these principles of neural coding might actually prevent certain types of neurofeedback learning from being successful. And so we test whether harnessing neural activity manifolds can actually guide more effective learning. So to do this, we designed a simple game where participants in the scanner used a joystick in order to navigate an avatar over to a goal. When this avatar reaches the goal location, so this flag, they get a 6-second break until the next trial. We then introduce a framework for manifold-informed neurofeedback procedures. So overnight, we take all the data and pre-process it from a subject performing this joystick task in the scanner. And we extract data from a large data-driven network of regions that are involved in navigation. We pre-process all of this data in order to fit a T-fate manifold, which is a nonlinear approach designed for the spatiotemporal conditions of single subject fMRI data. And we train a neural network model to be able to quickly extend the T-fate manifold in order to later embed new points into this same latent space during our real-time framework. And there's a lot of technical bits to unpack here, so if you have any questions afterwards, I'm happy to go through it more. So from this manifold, we then set our experimental mappings to determine how the manifold actually relates to the avatar's movement on screen. So for every data point, here we can consider the purple star. It gets a loading onto a given component of this manifold, which is then mapped to a direction of movement in the game. And if I were to then select a different data point in this latent space, it would result in a different movement vector in the game. And we can define a couple of different types of mappings over this manifold in order to test questions about the relationship between the manifold and learning in the game. So we set 3 different mappings that each kind of correspond, based on the neuroprosthetic literature, to components that are on manifold and off manifold, where the on-manifold ones, or the intrinsic and within manifold components here, are easy and kind of easy to learn, but we would expect an off-manifold component to be harder or impossible to learn. So to run this study, we use a closed loop system where our MRI scanner acquires a new brain image every 2 seconds, which is transmitted over to our cluster, where we perform some pre-processing and embedding onto that pre-trained manifold space. Still within this 2-second window, the value of this mapping is returned to the display computer, and it's used to update the avatar's location, or the avatar's turning direction in space. And this game just runs continuously so that subjects aren't getting dizzy from the constant starting and stopping of the avatar, but its turning direction is only updated once per volume. And you'll get a taste of what this actually looks like in a moment. This is a 4-session fMRI experiment where subjects return to the scanner for 4 to 5 consecutive days. In session 1, they perform this task with a joystick, and we do all the overnight training. In session 2, we introduce neurofeedback, and subjects are always trained in session 2 to control the avatar's movement based on the intrinsic or that easiest component. And in sessions 3 and 4, we test the within and the off manifold conditions to probe how the manifold actually constrains neurofeedback learning. So to introduce 2 additional manipulations after the easiest component is learned. And crucially, these orders are counterbalanced across subjects. So at each time point, we get a brain activation pattern, which maps to a direction in the game. And since this task is kind of hard and unnatural, the next step in the game is a weighting of the brain activity and a staircased confidence parameter, which we call brain control here. And this is altered at the end of every trial, depending upon a participant's performance. If they improve, it means they got over to the flag faster, or vice versa if they do worse. This parameter is staircased up and down accordingly. So we can use it as a behavioral metric of learning in this task. And based on this, we'd expect brain control to increase during the intrinsic and within conditions, but not in the off condition. Here I'm going to plot these learning curves with the change in brain control on the y-axis across all of the training trials within each session type. So in the IM condition, we see strong learning effects right from the start. In the WM condition, we see a reliable yet slower curve than the IM condition, and virtually no learning in the OM condition. And this is reflected if we just look across learning in all the trials within sessions of neurofeedback. And this is what it might actually look like in action. So these are real trials here. The information in black at the top is the display participants can see in the scanner. And their goal is to always level up, which means that they've increased in the amount of control they have. The white text at the bottom is just a demo. It's not shown to the participants, it's just for us. It counts the volumes that are coming in. And so if you watch in this trial, you can see that each volume results in a different decoded angle. So the avatar is turning in a slightly different location. And it's slightly biased back towards the center because it only has 80% of the control over the avatar's movement. But just to show you that in contrast with an off-manifold component, so let's look at the final run of a session in the off-manifold condition if we were to artificially set this parameter to match the IM condition. So the avatar goes veering off in circles, and it shows that the participant hasn't actually learned to control this axis in the same way it had in that intrinsic condition. So based on the behavioral findings that we saw, we'd expect there to be a similar pattern of changes in neural activity within each of these conditions. So here I'm just going to plot a change in the neural variance that's explained by each component of the manifold at the end of its training relative to the start of the training. We find that the intrinsic component explains significantly more variance at the end of the session, as does that within manifold component, but the off-manifold component does not. And these significant results would correspond with our behavioral results. So last, we consider the relationship between these two metrics, between the behavioral and the neural learning effects that we saw. In the intrinsic condition, though we saw learning both in the brain and in behavior, we weren't certain if we'd actually see a strong relationship between these two variables because it's already the most natural component and it's being trained before either of the two other conditions. We actually don't see a significant relationship here, although both are pretty high. Neither predicts the other. And in the OM condition, we also don't see a relationship because learning doesn't really occur. However, in the WM session, in that within manifold session, we see that participants who can successfully realign their activity with the component actually do significantly better at the task, meaning that after learning the IM condition, now relearning to control this second on-manifold component requires a type of realignment that is actually possible within one training session. So, take-home points. We introduce a new framework for manifold-based real-time neurofeedback, which affords learning curves that require less training time than typical neurofeedback studies. And it indicates an important innovation for the types of manifold learning algorithms that are used for human neurofeedback work. Prior work in this space, so using manifold constraints on BCI learning, has really focused on motor cortex, but we show the same effect in higher order cognitive regions. And this work holds important implications for neurofeedback interventions, as well as for the types of dimensionality reduction that can be used to identify these target signals. And this is just a slice of the results that we have from this study, so stay tuned for our soon-to-be preprint on this. And thank you to my lab and my wonderful collaborators and everybody for sticking around for this talk.
I can ask a quick question. I was wondering, so maybe I missed this when you said, can you hear me? Okay. Maybe I missed this bit, but so can you go back and see which brain features are most relevant in learning this in manifold orientation? Like, did you say something?
So which features of the manifold
we're in the brain.
Where in the brain?
We're in the brain, yeah.
Okay, I was hoping somebody would ask me this question, because this is a result that I didn't have time to put in. But so we are targeting this kind of network of navigation-related regions. This was a large neurosynth search for navigation, and we took a pretty big ROI here. But actually, when we look back, well, first we did one analysis where we moved a searchlight through this entire mask in order to find the exact regions within the mask that were most affected by the neurofeedback. We found it across the whole mask. However, if you look, can I show navigator? When we looked back at the regions that showed improved decoding after neurofeedback as opposed to at the start of neurofeedback, we didn't find any regions that really significantly improved in their ability to do decoding. This was a searchlight-based decoding procedure. We didn't find any regions in the IM or in the OM conditions that showed improved decoding, but we did find improved decoding in the WM session. So this is looking at the end versus the beginning, and these voxels are after cluster-based thresholding. What's their bad significance here? And crucially, only 1% of these voxels overlap with the ones targeted in the neurofeedback.
That's cool. So. Okay, well, thank you very much. Let's thank Erica again. And before you guys take off, we're going to have a community meeting, I guess. But before that, I just want to say a huge thank you to Talia, my organizer, who really did most of the work, and to our Meeting Perfect team, who were incredible. And this has run smoother than I could have imagined a conference running, especially Shawnee, Lynn, and Jeff. Thank you, guys. Wait for it. And please, another thank you to the organizing committee, including critically Nico Shuk and Lorg Williams and others, our junior helpers, all of you guys, especially Greta and Jacob, and there was somebody else. Oh, and the sponsors who made it possible to have this in the expensive city of Boston. Okay, so thanks so much, and I guess we'll have a community meeting now. Okay, thanks, guys.